Despite the success of antiretroviral therapy, viral rebound is inevitable in almost all individuals when treatment is interrupted. The main obstacle to HIV cure is the persistence of HIV despite antiretroviral therapy and plasma viral suppression to undetectable levels on commercial assays. This talk will involve quite a lot of basic science, and I hope that you'll stick with me because I think that understanding the mechanism of HIV persistence is essential for the development of strategies to induce HIV remission. So in the presentation, I will cover the detection and quantification of HIV persistence, establishment of HIV persistence, the effect of art on HIV persistence, and mechanisms of viral persistence on art. So I'm sure you're all familiar with this, but I will just give you a quick reminder so that we can interpret the ongoing data more easily. So here you can see is HIV life cycle. HIV binds to CD4 and the co-receptors and gain entry into the cell. After HIV gains cellular entry, HIV RNA is reverse transcribed into viral DNA. Viral DNA then enters the nucleus and is integrated into the host DNA. Normally, when there is productive infection, the provirus will be transcribed using host machinery to a viral RNA, leading to viral protein production and virion formation. In rare instances, latency occurs, and this is characterized by a provirus that is by definition transcriptionally silent. So this is one of the recurring concepts that I will bring back throughout the talk, and that it's HIV persistent on art is due to the existence of firstly, the latent viral reservoir, and as I mentioned, the reservoir is made up of infected cells that harbor HIV DNA, that during latency, these cells are transcriptionally silent, so they do not make viral RNA or viral proteins, but they later can become activated and produce infectious HIV virions. Secondly, there is also ongoing transcription and virus production, and I'm quite careful to not use the word new rounds of infection in this context. So this figure illustrates the different sources of HIV persistence. So up here you can see the cell, the viral DNA or the provirus is in red, it's integrated into the host DNA. And this is what we typically think of a latently infected cell, in that it does not produce any viral RNA or viral protein. Next, you can see this cell is transcribing, so there's viral mRNA being produced. The production of mRNA does not necessarily imply a productive infection. Then you can see this cell is producing viral protein as well. And then finally, this cell is producing virions. And under the influence of ART with suppression, an infection of another cell is unlikely to occur. So after these broad concepts, I'll go into more detail, firstly on detecting and quantifying HIV persistence. So there are many methods to quantify the HIV reservoir. I'll just highlight the most commonly used methods that we tend to see in the literature. So firstly, the viral outgrowth assay. So the viral outgrowth assay is thought of as the gold standard to quantify the latent HIV reservoir. So in this assay, resting CD4 T cells are isolated from PBMC. They're plated in limiting dilution or at different concentrations, and then they're activated to induce viral transcription and virus production. Donor CD4 lymphoblasts are then added, at different time points to propagate the infection. The levels of activated virus released is measured by P24 assay by ELISA traditionally. The frequency of infected cell in the original sample is then estimated by Poisson statistics, and it is expressed as infected units per million. The other method to quantify the latent reservoir is the HIV DNA assay, where a single region of a conserved region, usually GAG or POL, is amplified by PCR and detected. Finally, something that's in the middle of the two, which is the tilde assay or the TET-REV-induced limiting dilution assay. In this assay, cells are also activated to induce HIV expression. And the evidence of replication competent virus is and it's indicated by the presence of TET and REV RNA, which are measured by a PCR method. This step do away with the need for virus propagation. So what are the advantages and disadvantages of each assay? The Q4 assay involves high number of cells. So you can see that cell numbers required are about 100 million PBMCs. This is a lot of cells, and this means that evaluation in tissue will be very difficult to get enough cells to quantify viral outgrowth in tissue. The HIV DNA, however, requires a much lower number of cells. There's also a major difference in the labor intensiveness of the two assays, with the Q4 taking 2 to 3 weeks to perform, and HIV DNA can be done in a run in a day. The main advantage of the Q4, of course, is that detection of virus indicate that these are replication-competent proviruses. Whereas the HIV DNA, because it just measures HIV DNA, there is what you measure are not necessarily viruses that are capable of setting up productive infection. And in fact, 98% of the viruses or proviruses that you measure on the HIV DNA assay is actually defective and are not capable of new rounds of infection. So you can see in this figure from Yachi-Ho in 2003, this is what our current assays are doing. So in yellow is the Q4 assay. Because that it only measures viruses that are induced after one round of infection, you tend to underestimate the reservoir because there are cells that are replication competent and they're intact, but they are not induced after one round of activation. So your reservoir is at the pink level. The Q4 underestimates it, and the HIV DNA, because it measures defective viruses as well, overestimates it. And you can see that the difference are measured in logs. More recently published in Nature in 2019 is a new assay, the intact proviral DNA assay, and there's a lot of interest in this assay because it is a droplet digital PCR assay. It detects intact cell-associated full-length genomic HIV DNA. So theoretically, it can differentiate deleted and hypermutated proviruses from intact proviruses because it uses 2 amplicon and a hypermutation discrimination probe. So this offers a lot of added advantage over the HIV DNA because of the ability to differentiate defective proviruses. So this is a very promising method to quantify the latent reservoir. As I mentioned before, there's also ongoing transcription as well as viral production going on whilst on ART with plasma viral suppression on antiretroviral therapy. So how do we detect those? Well, we can look at those through HIV RNA, and we can look at cell-associated HIV RNA. The presence does not necessarily imply productive infection. Or there is the single copy assay that measure plasma-free cell-free RNA or virions. And it is important to remember that the majority of individuals on ART with undetectable viral load on conventional commercial assays still have detected HIV RNA on single copy assays. So how is HIV persistence established? So the HIV reservoir is established very early. These are data from a study led by Dr. Jinternet and Voronich and Dr. Don Colby, who's in the audience. And it involves 8 participants who initiated antiretroviral therapy during FEBIC1 acute HIV infection. By definition, FEBIC1 infections mean that they're nucleic test positive, but P24 and antibody negative. And it is estimated that the infection occurred in less than a week. So you can see these individuals are started on ART extremely early, within a week after HIV infection. And they've been on ART for 2.8 years, and before the actual treatment interruption, the single copy assay was less than 0.45 copies per mil. The HIV DNA level prior to treatment interruption was at a median of one infected cell per million CD4 cells. So extremely, extremely low reservoir. However, viral rebound occurred in all 8 of these participants at a median of 26 days. So this study really hammers home the fact that though viral replication is stopped extremely early, the reservoir is already seeded. And I think most of you will be familiar with this. That is, the majority of the latent reservoir is in the memory CD4 T cell population, and this accounts for over 90% of the cellular reservoir. There has been lots of interest in other cell types that may propagate the reservoir, and a lot of interest has been in monocytes, tissue macrophages, and dendritic cells. And this is because these cells are also susceptible to HIV infection. They express CD4 and the co-receptors, and they're very long-lived. These cells have been shown to harbor HIV DNA in the setting of plasma virus suppression on ART. And there's evidence that HIV transcription can occur in the setting in macrophages with the production of HIV RNA as well as viral proteins in terms of P24. However, the contribution to the latent reservoir is still unclear at this stage. So what is the effect of ART on the latent reservoir? Here you can see the initiation of ART at time 0 in both studies. One study uses measurement of HIV DNA, and the other study using the Q4 with latently infected cells per million CD4. Here you can see in red are total copies of HIV DNA in CD4 T cells. So it's about 7,000 prior to ART initiation, dropped rapidly to 1,000 in a year's time, and then to 500 at about 4 years, and then subsequently remain very stable. So the reduction is about 90%, and this is grossly different from what we see with HIV RNA, where we see 5-6 logs of reduction within 6 months to a year. Similarly, using the Q4, you can also see that after ART initiation, especially after the first year, the decay of the reservoir is extremely slow. So the reservoir is very, very stable. The half-life of the reservoir is estimated to be 44 months in individuals on ART with plasma virus suppression. So this means that 73 years of ART is required to eradicate a reservoir of 1 million cells. So these are data from individuals who initiated antiretroviral therapy during chronic HIV infection. And of course, in our cohort, we're particularly interested in acute HIV infection. So these are data from studies led by, again, Dr. Jinternet and Voronich and Dr. Merlin Robb, from MHLP involving 2 different cohorts, both in Thailand, and the RV217 cohort also involved sites in Africa. And here you can see the 2 studies, the RV217 study, where participants are not initiated on antiretroviral therapy, and the RV254 study, where participants are initiated on antiretroviral therapy, usually within 1 or 2 days after the diagnosis of acute HIV infection. So what I want to point out is that prior to ART initiation, both cohorts had similar level of HIV DNA. Now, without treatment, the HIV DNA increased very rapidly, and then it really plateaus and doesn't change much. With ART initiation, you can see that there's a rapid drop in HIV DNA that reached to very low levels, and the divergence between the treated and untreated group occur early. So even though, as I mentioned before, the early ART does not eliminate the reservoir, however, it does reduce the size of the reservoir. So now I'll home in on what really happens to the reservoir after ART initiation. So what tissues can we identify HIV persistence? Basically, we can find HIV in all types of tissue in the body. The most common, of course, and the majority of the reservoir resides in secondary lymphoid tissues, including lymph node and lymphoid tissue in the gut. But HIV persistence has also been evident in the CNS, in the lungs, in the kidneys, in the reproductive tract, and even adipose tissue. So, as I mentioned before, the sources of HIV persistence can be latently infected cells. And this is because latently infected cells are long-lived. And so to target these cells, potentially we may use the kick and kill strategy for the kick to activate virus and kill mechanisms involving immune mechanism to get rid of the expressed virus. However, it's not as simple as that. There's also clonal expansion of latently infected cell. So this is one latently infected cell, but due to homeostatic proliferation or antigen stimulation or integration in genes that control growth and development of cells or cancer-associated genes, these cells actually proliferate. So you get more and more latently infected cells. And these cells, importantly, have identical HIV sequences and the same integration site. And this is because they're from the same clone. So clonal expansion is actually very important. It accounts for 50% of the inducible replication competent HIV reservoir. Clonally expanded virus can be intact and infectious, and these clones have been shown to be a source of initial rebound viremia during analytic treatment interruption. Now, so how do we get rid of these clones? Potentially, anti-proliferative therapy may be useful, and there's certainly clinical trials going on at the moment that looks at the effect of mycophenolate in reducing proliferation and targeting the clones. And as I mentioned before, 80% of participants on ART still have persistent viremia measured by single copy assays. So what are the sources of these then? These are obviously not latent by definition because there's ongoing transcription. So what's going on? Well, the potential sources are ongoing cycles of viral replication and infection. This is quite controversial. It may be due to inadequate ART potency or inadequate penetration at sanctuary sites. The other possibility is ongoing viral production from integrated proviruses, but ongoing cycles of infection are blocked by ART. So you may think, well, why is it important? Like, why do we need to go into all these details? What's the point of understanding this? Well, I think it's important because the ways of targeting these mechanisms are very different. So if the ongoing cycles of viral replication and infection is due to inadequate ART potency or penetration, then the focus should be to improve the ART. However, if the ongoing virus production is from clones of integrated proviruses, these proviruses are not targeted by the existing immune responses. Furthermore, latency reversal agents will also not affect these populations because these cells are not latent, they're already actively transcribing. And the question is raised of, well, whether we can use block and lock strategy of locking these cells so that they're not transcriptionally active. And of course, block and lock strategies are at very early stage development. They're only in animal models at the moment. So going back to ongoing cycles of viral replication and infection, if there's ongoing cycles of viral replication and infection, you will see that there will be mutated sequence because of the error-prone HIV reverse transcriptase, and the provirus will be integrated at a different site. So this is in contrast to clonal proliferation. Ongoing cycles of viral replication and infection is unlikely in the peripheral blood, and this is because of data that shows that emergence of new variants whilst on ART is rare, and the majority of studies shows that no reduction in the level of persistent of viremia with treatment intensification. However, this may occur in sanctuary sites, and two sites that I want to point out are the lymph node and the CNS. And this is because both sites have evidence of reduced ART penetration. In the lymph node, it's been shown that in the follicular area, there's reduced immune-mediated killing. In both areas, we can find HIV RNA expression despite suppressive ART in peripheral blood. And finally, there's evidence of sequence evolution on ART. So it may be possible that there's ongoing viral replication in sanctuary sites, but we don't really see much evidence of that reflected in the peripheral blood. So the second option is ongoing virus production from clones of integrated proviruses. So it's been shown that clonally expanded intact proviruses can produce sufficient virus to cause detectable low-level viremia. And in these scenarios, in these particular patients investigated, the plasma viral sequences or viral RNA matches the proviral sequences, so the HIV DNA sequences, as well as matches the QFOA well sequences. So what does that mean? Well, that sort of translates to the fact that clonal proviruses are replication competent. They can be induced in the QFOA to produce viruses, and these viruses are reflected in mRNA, matching mRNA sequences in the blood. Importantly, in these studies, it also suggests that new rounds of infection are not set up on ART, and this is because there's no evidence of ART resistance mutation. There's also no evidence of sequence evolution in the clones, and the important question is, well, how are these viruses being expressed and produced and circulated in the blood without any immune elimination? And how do we suppress these clones? So these questions, really, we do not have answers and are important areas to research in. So in summary, I think that it's very important to understand the mechanisms of HIV persistent in the development of strategies to induce HIV remission. And I think I've covered in my talk that the HIV persistence on ART is due to the existence of firstly the latent reservoir, but there's also the element of ongoing transcription and virus production as well. And I would like to acknowledge a number of people from MHLP, Dr. Chintanet Anamornich and Dr. Sandhya Fasan, who are my mentors and supervisors, Dr. Eugene Kroon for his help in the presentation, and also the participants in the MHLP studies of the RV254 and RV217, and the collaborators involved in those studies. Thank you very much.